So guys, in this, let us look at this question. Explain the formation of primitive streak and its fate. They have asked us for 5 marks. Explain the formation of primitive streak and its fate. So first of all, did you understand which topic it is? It is embryology. Okay. So now let's get started with this. What is this primitive streak? How does it form and what is its fate? Okay. So basically this primitive streak is a narrow linear groove. Okay, which you can see here, it is a narrow linear groove which is formed in the epiblast. So this blue surface, whatever you're looking at, is the epiblast. And where does it form? It forms at the caudal end, that is the tail end, okay, and in the middle, exactly in the middle. So the medial plane, okay, a caudal end, median plane, dorsal surface at the back of what? Of the bilaminar germ disc stage embryo, around the 15th day from fertilization. So when the fertilization has occurred and it has implanted and all that, okay, the day 15, you should be able to see this primitive streak. And where is this primitive streak forming? In the epiblast. Where in the epiblast? In the caudal end. In the caudal end where? Exactly in the middle. And in okay, that is what, okay. It is the dorsal surface of the bilaminar germ disc stage embryo. Too much information, but we need to revise now. Tell me, what is primitive streak and how does it form? It forms in the bilaminar germ disc of the embryo in the dorsal surface, that is in the epiblast caudal end, medial plane. It is a narrow linear groove. Narrow linear groove, epiblast, caudal, medial plane, dorsal surface, bilaminar germ disc stage embryo, 15th day from fertilization. This is how it forms. Now we will try to draw it. Otherwise, if without diagram, you will not get marks, right? First of all is a diagram. It will also put your thoughts into perspective, right? So now let us see. This is our embryo. So this part you can remove. Yeah. Okay. Now around this you draw 2 more layers. Okay. That will be, one will be the amniotic thing and the other one will be the yolk sac. I'm thinking around it completely you draw these 2 more lines. Okay. Now let's draw the primitive streak, guys. Get ready. This is the main part of the exam, right? This is the primitive streak, right? So beautiful. See, it's like a keyhole to me, but actually it should be more longer and narrow, I think. Okay. In the middle, what you will give them? One pit, and you will give them one primitive groove. Okay, now let us label, and here you will see that this is the 15th day from fertilization. This is the embryo, bilaminar germ disc stage embryo. Okay, day 15th day or day 15. Okay. Now, what else you will say? This is the oropharyngeal membrane, future mouth. Okay, and this one is going to be your primitive pit. Inside it is that whole primitive pit. Around that, the raised part is the primitive node. So node is around the pit. Then this one inside, whatever is there, that is a primitive groove, that narrow inside, that depression. And around it, that raised part will become the primitive ridge. Okay. Entire these 4 together you can call as what? Primitive streak. Okay. So these terms are very important. Primitive node, primitive pit, primitive groove, primitive ridge. Okay. So where is the primitive node? Primitive node is cephalic. It's more towards the head side, right? What is it around? Primitive node. Node is around pit, right? And ridge is around groove. This much you have understood now. So you were drawing those 2 layers. You remember what is that amniotic, what is that yolk and all that? We'll see that now. So here you can see that this is the blue color is the epiblast and the yellow is the hypoblast. Below the epi is the hypo and above the hypo is what? Epi. Very good. And the primitive streak is forming where? In the epiblast, in the caudal end, medial plane, on the dorsal surface, right? This is where the primitive streak is forming. So when you drew it, we just showed that above this, all this amniotic cavity is there, and below this epiblast there is hypoblast, and below which you have the yolk sac. Okay. Okay, very important. That is why we drew those two extra lines around that entire thing. In this also they have drawn those two extra lines, right? Okay. Now why are they asking us to learn all this in the exam? That we should, that's what they ask, right? See, now when you look at this embryo, you know which the cranial end is going to be and which the caudal end is going to be, correct? So that is one thing. It's not that it is decided at this point, I'm thinking, but it is, once you look at it, you will be able to tell clearly, okay, this is the cranial end, this is the caudal end. And the laterality is established. See, the center line you know. Now, if you know center line that is medial, definitely you know which is lateral. Correct? Laterality is established. Then cardiogenic area, the heart. Where will the heart form? You know, heart is forming actually somewhere here. They have shown this as cardiogenic area, but actually it is above the mouth in this image. Remember, this is dorsal surface and mouth is actually ventral. Okay, so you just understand that's why they put dotted line, I think, here for the precaudal plate, future mouth oropharyngeal membrane. Okay, now coming to the most important thing. What is this primitive streak? What does it indicate? It indicates the beginning of gastrulation. What is gastrulation now? Gastrulation will make a 2-layer thing into a 3-layer thing. So that is what is gastrulation. And this primitive streak is required for this process of 2-layer becoming 3-layer. So this entire thing is what? Gastrulation. 2-layer becoming 3-layer is gastrulation. And what does it need? It needs the primitive streak. Why? Because the epiblast cells migrate through the primitive streak to form the endoderm. What is that? Now that is what is the fate of all this also. Okay. So now look at this. Gastrulation is the process of forming 3 primary germ layers from the epiblast. Okay. See, it is not from the 2 it is becoming 3. From one of the 2 only it is becoming 3. See, let us understand this. Again, 2 is becoming 3 is correct. So you have epiblast and hypoblast. Which is the 2 layers. Now it is becoming ectoderm, mesoderm and endoderm. But all these layers are actually coming from epiblast only. Okay. So these epiblast cells will go through the primary streak and form the 3 germ layers. Okay. So gastrulation is the formation of 3 primary germ layers from the epiblast. How? By the epiblast moving through the primitive streak. So now look at this diagram here. So the primitive, these epiblast cells are in blue, right? That's what they're showing. They migrate, they go into this primitive streak, and then what they do, they become some flask-shaped cells or something, and then they go and replace the hypoblast cells. Hypoblast destroyed. Poor guy. Hypoblast gives away, and whichever epiblast goes and sits in the hypoblast's place, they become what? Endoderm. Inside, they're going inside, right? They become endoderm. Hypoblast replaced by epiblast cells, now called as what? Endoderm. And the one who is sitting here between the epiblast and the endoderm or the hypoblast, that is the mesoderm. Mesoderm. And whoever doesn't move, I don't move at all, they become the ectoderm, your brain, ectoderm. Okay? Got it now? Very good. So this is what is the fate of the all these epiblast cells actually. I wouldn't say it's fate of primitive streak. It's the fate. Okay. Some terminologies that you have to use in the exam. Let us look at this. Migrate word should be there. They become flask-shaped cells. They invaginate into this primitive streak. Then there's some things that control all these genes and all. Okay. All this much is enough, I think. So the epiblast is the one source for all the 3 germ layers, and those 3 germ layers only form our entire body, that is the tissues and organs. Is it clear now? Easy? In the end, you write one clinical anatomy, okay? Tumors associated with gastrulation, okay? So what is there here? The remnants of primitive streak can form tumor, sacrococcygeal teratoma. Remnants of primitive streak. Okay. Another condition is the sirenomelia. Sirenomelia or mermaid syndrome. Insufficient production of mesoderm by the primitive streak. The legs are fused, guys. So it will give that mermaid kind of effect. And here you should understand that these people are saying that it is not at all formed correctly. It is genesis or agenesis or say caudal agenesis or sacral agenesis. Okay, they are saying it is these are the better terms. Okay, actually it is found more in infants of diabetic mothers. So this is very important to know. Its cause is not known, but it occurs more frequently in diabetic mothers. This is where we have to focus. Infants of diabetic mothers. Okay, the caudal agenesis. I'm sure you would have studied. Okay. Just another thing, guys. If that heart, the cardiogenic area, we can define something like that we studied, right? So basically, the two things will come: primary heart field, okay, and then secondary heart field. Okay, two things will come. I think secondary they have not shown here. So two things get defined. Okay, that is how you get the cardiogenic area. Here they have shown the cardiogenic area, if you remember. Okay, so importance of the primitive streak and all when you studied. Okay, so now you have studied this question, formation of primitive streak and its fate. Okay.